Bismillahirrahmanirrahim. Assalamu alaikum, second year. How are you? I hope you will be fine. Today we will start our chapter number 21, Development and Aging. And this is lecture number 1. After the completion of this chapter or this lecture, you will be able to describe the cleavage and relate it with amount of yolk. Explain the events of gastrulation, list the tissue and organ formed from the 3 germ layers, state the events of neurulation, formation of neural crest, and list of the structures that are derived from neural crest cells and define organogenesis. Development and aging. Now, what is development and aging? It is a period of extensive growth in size and mass as well as ongoing differentiation of organ system established in embryonic period. Different systems formed as brain, respiratory system, digestive system. These are positive changes which lead an organism from its zygotic stage to adult form, and then further growth leads an organism to its full size. These positive changes become negative at some stage in the life are termed aging. Embryonic development, the progressive changes which are undergone before an organism acquires its adult-like form or shape, constitute the embryonic development. It begins with a series of mitotic division in the zygote, which leads to a multicellular stage called the embryo. Finally, an adult-like body is formed. The study of an organism at this stage is called embryology. Now we have different stages of embryonic development, and we can divide these stages, or we can divide the embryonic development into 4 categories or stages: the cleavage, gastrulation, organogenesis, and growth. The first one is cleavage. Following fertilization, the zygote undergoes a mitotic division called the cleavage. Morula. The first division results in the formation of 2 identical cells called blastomeres. DNA replication and mitotic division occur repeatedly. The cells get smaller and smaller with each division. Finally, a solid ball of small cells, the morula, is formed. The morula is still about the same size as the zygote. This is the zygote, and this morula is the result of this cleavage. Blastula. As cleavage or division continues, cells begin to move apart so that space appears among cells in the center of mass. Cells keep pulling away from the central area, forming a fluid-filled cavity known as blastocele. You can see here the zygote, cleavage formation, 8-cell stage, then blastula, and here is the cavity which is known as blastocele. This hollow sphere embryo which develops at the end of cleavage is called blastula. Now at this stage, the zygote is known as blastula within a cavity that is known as blastocele. Blastocyst. This embryonic stage with blastula in mammals is called as blastocyst. Now again, what is blastula? With a cavity, blastocele, the zygote is known as blastula, and the embryonic stage of blastula is known as blastocyst. This is a fluid-filled hollow sphere composed of a single layer of large flattened cells called trophoblast cells and a small cluster of 20 to 30 rounded cells called the inner cell mass located at the one site. This is blastocele. Now it is blastula, blastocele, and these are the different stages of embryonic development. Here we have a video to understand the cleavage formation and blastocyst formation. The quantities of yolk in egg. Many animal eggs contain yolk. It is a mixture of proteins, phospholipids and fats. The amount and distribution of yolk vary among animals. Most invertebrates and simple chordates have egg with relatively small amount of yolk uniformly distributed through the cytoplasm. Many vertebrates have large amount of yolk concentrated at one end of the cell and known as the vegetal pole. The opposite pole is called the animal pole. Different patterns of cleavage based upon amount of yolk. The amount of yolk in the egg affects the pattern of cleavage. So we can divide the patterns of cleavage into two types, holoblastic and meroblastic. Now, what is the holoblastic type of cleavage? In this type of cleavage, egg with evenly distributed yolk, cells of roughly same size, bony fishes and amphibians are the examples of holoblastic cleavage. Meroblastic cleavage, very large amount of yolk. Cell division takes place only in the blastodisc in one side of the cell, and this type of cleavage is found in reptiles, birds and some fishes. Now the second step or the second part of the embryonic development is gastrulation. Second phase, characterized by the differentiation of embryonic germ layers. Embryo is transformed from blastula into a three-layered the gastrula. These three layers produced from embryonic tissue called the ectoderm, endoderm and mesoderm. Ectoderm is the outermost, endoderm is the innermost and mesoderm is in between these two. Gastrulation varies somewhat depending on the species. Here we will focus on the gastrulation in humans. It begins about the 15th day after fertilization. The trophoblast thickens at one point to form a mass of cells called the inner mass cells. After implantation, as you seen in the previous video, the inner cell mass grows and splits, forming two fluid-filled sacs that are separated by a double layer of cells called the embryonic disc. One sac is amniotic sac, which is bounded by the amnion and filled by amniotic fluid. The other sac is yolk sac, but it does not contain yolk as human embryo takes nutrition from mother's body. Gastrulation. At this stage, the embryonic disc consists of an upper layer and lower layer. The upper is called epiblast and lower is facing the amniotic sac, and which later on develops into ectoderm and mesoderm. And then the lower layer is called the hypoblast, and on the side facing the yolk sac, which is in future form the endoderm. The upper and lower layer split apart slightly and slit the primitive streak corresponding to the blastopore appears in the center of the upper layer. You can see here in the figure gastrulation, the upper and lower layer. The early gastrula, you can see the ectoderm. This is the outer ectoderm. This is a endoderm, and here we have the blastopore, and inward movement of cells is the result of mesoderm. Outer ectoderm, then endoderm, and blastopore formation, and inward movement of blastopore formation, the blastopore is the result of formation of mesoderm. So here we have the outer ectoderm, endoderm, and this is the mesoderm from the place of blastopore. The upper layer is now ectoderm and inner is endoderm. The cells of the ectoderm migrate through the primitive streak into the interior of the embryo, forming the mesoderm. Here we have another view for the formation of 3 germinal layers. We will revise all these processes of cleavage. Zygote, then cleavage formation, again the cleavage. From 8 cells we have a blastula, a ball of cells. Then the appearance of this blastula is like this. We have a blastocele, okay, and this is the cross section of blastula, and then the conversion of blastula into gastrulation, and in gastrulation now the inward movement of cells from and the formation of blastopore, and now this is the future ectoderm, this is future endoderm, and here is the formation of mesoderm. You can see here the blastopore, okay, finally formed, and this is the ectoderm, this is endoderm. Here we have the blastocele. Again you can see the blastopore in gastrula, and ectoderm the outer layer, endoderm the inner layer, and in between these two layers by the formation of inward movement we have the mesoderm. Fate of 3 germ layers or the germinal layers. Embryonic germ layers or the germinal layers, ectoderm outer layer, mesoderm middle layer, and endoderm inner layer. By ectoderm, the formation of epidermis of skin, epithelial lining of mouth and rectum, and nervous system. And mesoderm is involved in the formation of skeleton, muscular system, dermis of skin, circulatory system, excretory system, reproductive system, including most epithelial linings, outer layer of respiratory and digestive systems. Endoderm is involved in the epithelial lining of digestive tract and respiratory tract, associated glands of these systems, and epithelial lining of the urinary bladder. Here we have a video to understand the gastrulation in the embryology.
By day 13, the blastocyst is firmly implanted in the endometrium. We'll now make sagittal cuts through each constituent part, one after another. The outermost is the syncytial trophoblast, which secretes the beta-HCG we detect in pregnancy testing. Next is the cytotrophoblast, followed by extra-embryonic mesoderm, inside of which is a space known as the chorionic cavity. Sitting in this cavity and also surrounded by extra-embryonic mesoderm is the developing embryo, the parts of which we'll discuss next. The epiblast sits atop the hypoblast. Together, these are known as the bilaminar disc. Cells of the hypoblast extend out to form this space called the yolk sac. By this stage, the epiblast has developed inside it the amniotic cavity. We'll remove the outer elements in order to take a closer look at the bilaminar disc. At At the cranial end of the hypoblast lies the procaudal plate, an area in which columnar cells are present. It serves as a marker of the embryo's cranial aspect. The end point of gastrulation is the formation of a trilaminar disc made up of ectoderm, mesoderm, and endoderm. The cells that make up these layers were all once of the epiblast. This transition is achieved as follows. Cells of the epiblast migrate inward, downward, and then differentiate. From either side, this continuous passage begins like symmetrical waterfalls. To be clear, it is the moving cells which produce this form, as shown here. Macroscopically, this makes a visible groove in the surface of the epiblast known as the primitive streak. Its presence marks the beginning of gastrulation. At the cranial end of the streak, cells are migrating thickly, forming this circular depression known as the primitive node. As the embryo grows, more cells migrate, the primitive streak elongates. Cells which differentiate into endoderm displace cells of the hypoblast and eventually replace it entirely. Cells which remain in the upper layer differentiate into ectoderm. Cells which differentiate into mesoderm force their way between these two layers. Around day 16, cells migrating through the area known as the primitive node begin to form a hollow rod which curves toward the cranial end of the embryo. It extends toward the procaudal plate and is known as the notocaudal process. We take a coronal cut now. We see the hollow rod, and if we move through it now, we should also see its connection to the primitive node. As the notocaudal process grows longer, the embryo continues to grow in all dimensions. Around day 18, the notocaudal process fuses with the endoderm. From this to this. It's now known as the notocaudal plate. Being continuous with the primitive node, this now allows free passage between the amniotic cavity and the yolk sac. Let's take a look through to solidify that, starting at the primitive node. We're looking down through what is known as the neuroenteric canal, and the space we can see into is the yolk sac. It's theorized that this brief communication between amniotic cavity and yolk sac allows pressure equilibration between the two cavities of the early embryo. It fills almost all the space between the ecto and endoderm. Apart from the space the notochord occupies, it is absent also at the oropharyngeal membrane, located where the procaudal plate once was, and the cloacal membrane. These are structural elements of the embryo that currently exist as ecto and endoderm tightly adhered. And that concludes the process of gastrulation.
I hope gastrulation is clear now. The organogenesis. The formation of organs and system during embryonic development is called organogenesis. Neurulation, differentiation of ectoderm, and production of brain and spinal cord. Here is the position of zygote, animal pole, neural plate or the nervous system formation, epidermis or the skin, and then the mesoderm, which is the future muscle, kidney, bone, and gonads. And this is endoderm, which forms the digestive organs and lungs, and this is the vegetal pole. Now the neurulation in human embryo. You can see here the notochord in the first figure and the ectoderm labeled here. Then the neural plate and formation of different portions of the germ layers are seen here. The ectoderm, mesoderm, and this is the notochord. Notochord forms from mesoderm cells soon after the gastrulation is completed. Then the again look at the second figure. Here we have the ectoderm, mesoderm, endoderm, and then the signals from notochord cause inward folding of ectoderm at the neural plate. Then in third step, the neural tube formation, and here is the endoderm and neural tube formation. The ends of neural plate fuse and disconnect to form an autonomous neural tube. Neurulation. Neurulation in human embryo. In human embryo, the process of neurulation begins during the first month of development, and the nervous system develops from the middle ectoderm located just above the notochord, as you can see in the figure. The developing notochord includes the induces or the stimulates the overlying ectoderm to thicken, forming the neural plate, which is seen along the dorsal surface of the embryo. Central cells of the neural plate move downward and form a depression called the neural groove. Then, neural folds develop on either side of the neural groove. By 22nd day, the continued cell movement bring the superior margins of the neural fold closer together until they meet and fuse, forming the neural tube. It soon pinches off and becomes covered by surface ectoderm. At this point, the embryo is called neurula, and the later the anterior of the neural tube grows and differentiates into brain. The remainder of the tube develops into the spinal cord. By the end of the first month of development, the three primary brain vesicles, fore, mid, and hind brain, are obvious. By the end of the second month, all brain flexures are evident, and the cerebral hemispheres cover the top of the brain stem, and brain waves can be recorded. Now, the role of neural crest. Here you can see the labelings of this figure. Here we have the ectoderm, then the dorsal edges of neural plate and neural crest. These are the neural crest portion. Neural crest again, ectoderm and neural tube here, and this is the notochord. Various motor neurons grow out of the developing brain and spinal cord, but sensory nerves have a separate origin. The neural crest, which is developed in the region of the neural plate border after neural tube closure. The associated neural crest cells migrate widely to the lateral sides of the neural tube and give rise to a cranial, spinal, sympathetic ganglia, and associated nerves. Neural crest cells subsequently migrate to various parts of the embryo, forming peripheral nervous system, medulla of the adrenal gland, teeth, skull bones, and so many other different cell types that some have proposed considering neural crest called as fourth germ layer. Here we have a video to understand the neurulation.
By day 16, the embryo consists of three layers: endoderm, mesoderm, and ectoderm. Ectoderm develops into the nervous system, and the first phase of this process is known as neurulation. Around day 18, part of the ectoderm begins to thicken. This portion is known as the neural plate, in dark blue, between the cranial end of the embryo and the primitive node. Neurulation is the process of neural plate becoming neural tube. Got a schematic animation here to try and illustrate that process. We'll break this down day by day throughout the rest of the video. On day 19, the neural plate begins to buckle in the midline. We call this dip the neural groove, and the mounds either side the neural folds. These folds progress toward each other and begin to fuse around day 21. Where they have fused, they form a hollow cylinder, which is the neural tube. Fusion then continues in synchrony toward caudal and cranial ends. The as yet unfused portions are known respectively as the cranial neuropore and caudal neuropore. Pore literally means minute opening. The minute opening at the cranial end closes around day 24. And the caudal end closes around day 26. A particular group of cells reside at the peaks of the neural folds. These are the neural crest cells, which form a layer over the top of the closed neural tube. These develop into most of the peripheral nervous system. To sum up, neurulation is the process of neural plate becoming neural tube. The closure of the neural folds begins in the center and zips up towards either end. The central nervous system is developed from the neural tube. It's worth mentioning that by this gestational age, the embryo has started curving into the fetal position. We've kept the ectoderm flat to keep our schematic version really simple, but these pictures from Gray's Anatomy accurately show the embryo at around 21 and 31 days. The neural tube curves through the back of them, like so. That's it for now. Hit subscribe if you like this video.
Here we have another video for neurulation. Please concentrate on this video. organogenesis. As you can see, the endoderm, mesoderm, and ectoderm, three germ layers. Endoderm involved in the formation of lung cells, thyroid cells, digestive cells, and mesoderm is involved in the cardiac muscle cells, skeletal muscle cells, tubule cells of kidney, red blood cells, smooth muscle cells in gut. Ectoderm is involved in the skin cells of epidermis and neuron